Okay. So David, I will introduce you in Spanish. I always do that in Spanish. And I always say that I promise I won't say anything mean or anything wrong. So
you seem like a very nice person, Eva.
Okay. Thank you. Okay. So, pues buenas tardes a todos. Gracias por unirse una vez más a nuestros seminarios de Frontiers in Genomics. Como les explicamos, este semestre las conferencias son en principio sobre todo en las mañanas debido a los horarios en los que se encuentran nuestros invitados, pero justamente hoy tenemos una excepción y empezamos a las 5 de la tarde, pues porque nuestro invitado está en Australia. Entonces, pues este era el único horario en el que podíamos encontrarnos todos juntos. Bueno, pues es un gran placer presentar y tener hoy con nosotros al doctor David Goode. Él trabaja en el Peter MacCallum Cancer Center en Melbourne, en Australia, y trabaja en el departamento de cáncer, particularmente en el departamento de genómica de biología computacional del cáncer. Su especialidad, como van a ver ustedes, es la genómica evolutiva del cáncer. Se graduó en la Universidad de Stanford y después de una serie de estancias en la Universidad de British Columbia y en el Walter and Eliza Hall Institute of Medical Research. También realizó un postdoc en el Peter MacCallum Cancer Center y ahora es investigador y líder del área de biología computacional del cáncer. Sus áreas de trabajo son realmente muy interesantes, como van a ver ustedes en unos minutos, y sus áreas de trabajo se centran en estudiar la evolución molecular del cáncer y estudiar a nivel poblacional y a nivel de célula única para identificar los mecanismos evolutivos que dirigen la respuesta de las células tumorales a los diferentes tratamientos antitumorales, ya sea terapias por radiación o quimioterapias. Y entonces para hacer esta investigación, es una investigación en la cual se combinan muchas disciplinas como la bioinformática, la genómica, la evolución molecular y laboratorios, o sea, técnicas obviamente en donde se generan datos por wet lab y muchas técnicas de análisis por dry lab también, ¿no? Como van a ver ustedes. Sin decirles más, pues bueno, pues quisiéramos agradecer. David, thank you so much for agreeing to participate for 2 days in a row in our program. And yesterday it was absolutely, we were all very, very interested and very enthusiastic about your seminar. And I'm sure that today we will be even more. And we have a lot of our colleagues that are here to listen to you.
Well, thank you, Yvonne. Thank you for that introduction, and thank you for inviting me to be part of your program. It's been a great experience so far. Yeah, I had a great time discussing cancer evolution with the students yesterday, and today I'm looking forward to telling you a little bit about some of the work my lab has been doing in that space. So I will share my slides now. And so can everyone, hopefully that worked. Everyone can see my slides now?
We can see your slides. That's perfect.
Great. Okay, cool. Well, yes, thank you everyone for coming today, or for tuning in today to listen to my talk. I'm going to tell you about, as I said, some of the research my group has been doing, and we use a technique called evolutionary genomics, where we combine sort of genomic analysis and transcriptome analysis with methods from evolutionary biology and population genetics to investigate how cancer forms and progresses, and in particular, how it evolves resistance to commonly used therapies. So I'll go into about 3 vignettes today on projects that are ongoing in my lab at the Peter MacCallum Cancer Center. So there's a picture of the Peter Mac, as we call it. It's Melbourne's main cancer hospital. And we're moved into this shiny new building recently where half the building is a hospital and half of the building is the research labs. And it's a really great environment to do research because you have sort of a lot of links between the basic and clinical research areas. So given it's a general audience, I thought I would just start out with sort of a basic overview of the evolution of cancer and how tumors form and evolve. And so what I'm showing here is kind of a representation then of this process. And what happens, tumor, most cells in sort of a normal, sort of stable, differentiated tissue, they only divide whenever and wherever they have to. But through mutation or other processes, cells can, individual cells may escape these controls on their growth and proliferation and start growing and dividing when they're not supposed to, and that can initiate tumor formation. So over time, this sort of aberrant cells, they expand and they proliferate and they invade the neighboring tissues and kind of take over a local area, and that's the start of tumor genesis. And through various processes, genetic mutation, but also increasingly sort of plasticity at the transcriptional and epigenetic level, the tumor evolves to become a collection of related but distinct subpopulations, here represented by the different colors. So each of these clones, as we call it, each of these subpopulations of cells will have their own genetic profiles, have their own gene expression profiles, and these will confer distinct phenotypes to each of these clones. And through the process of selection, certain clones will start to emerge. Certain clones will have better fitness under the sort of local environment that tumor is in. And there's a tremendous amount of selective pressure on tumors as they're growing and progressing. There's certainly a lot of competition for space and nutrients. Often oxygen levels will be low, as you get this sort of rapid expansion of highly proliferative cells. Often tumor cells sustain quite a lot of DNA damage as they grow. That's usually a big driver of tumor genesis is genomic instability. And you also have the host immune system that is trying to seek out and kill aberrant cells, such as cancer cells. So there are all these stressors on cancer cells. They have to be very good at adapting. They have to be very good at changing quickly to suit their environment. And so sort of with this process of the diversity that is created by mutation and plasticity, plus the selection that is imposed by sort of internal and external factors, the tumor evolves over time and its composition will change. And so in this example, maybe these yellow cells actually best adapted to the local environment that tumor finds itself in. And so over time, they become the predominant clonal population in that tumor. And as well, through the process of metastasis, cancer cells can spread to other parts of the body where they can seed new tumors, and those tumors will have their own evolutionary histories, their own evolutionary trajectories, and new clones can arise at these distant sites. And so then, at some point, when the tumor reaches a certain size, that will cause the patient some discomfort, and they will go and see their doctor and be diagnosed with cancer. And at that point, then they'll undergo some sort of treatment. That'll be some combination of surgery, radiotherapy, or chemotherapy. And that may be successful in the short term in knocking out the tumor at the primary site, so the site where the tumor originated. But there can be these metastatic deposits where the tumor live on and in fact evolve again in response to therapy. And so in this example, these purple cells that formed at this metastatic site have some kind of intrinsic resistance to the treatment that was given to this patient. And so they come back and they reseed the tumor at this distant metastatic site. And eventually that patient would relapse and return to the clinic with a tumor that's actually molecularly could be very different from the tumor that patient originally presented with. And so all of this occurs through this evolutionary process, as I mentioned, where there's a high level of diversity, there are strong selective pressures, and the tumor changes over time. So my group is really interested in understanding these evolutionary processes in cancer, how they shape tumor formation, and how they influence response to treatment. And as I mentioned, we do this with a process we call evolutionary genomics, where we do genomic and transcriptomic profiling of tumors from both patients and model systems. And we look at how tumors change over time. And we also do evolutionary analyses to understand how tumors form and how they grow. And so I'm going to show you today sort of 3 projects that we have that cover kind of different time scales of tumor evolution. So the first one that I'll show you today is actually concerned more with the sort of initiation and progression of tumorigenesis. And whereas a lot of evolutionary studies in cancer are kind of the microevolutionary level, where you're comparing sort of samples from the same patient over time, this project actually takes a bigger sort of macroevolutionary view of cancer as a consequence of the breakdown of multicellularity in differentiated tissues. And I'll get into more about how that works shortly. The second project I want to talk to you about today is where we do computational reconstruction of tumor evolution, where we actually simulate the growth and evolution of tumors in silico to then try to understand what sort of important evolutionary events in terms of emergence of new clones and selection on those clones, what types of important events are occurring early in tumor evolution that shape the properties, eventual properties of the tumor that forms within a patient. And then finally, I'm going to show you some work we're doing with patient-derived xenografts, looking at how patient tumors change pre and post therapy in prostate cancer, sort of what transcriptional changes and genetic changes are maybe driving resistance to androgen deprivation therapy in prostate cancer. So those are the 3 topic areas I'll cover today, and go into each of those, sort of give you an overview and a flavor of each of those projects. Excuse me. So the first one I'll talk to you about today is about the disruption and rewiring of metazoan gene regulatory networks in cancer. And this is work that has been performed by my former PhD student, Anna Trigos, who's now a postdoc at the Peter Mac, and has been picked up by a new PhD student, joined the group, Mikhail Diaz. We collaborate with Professors Rick Pearson and Tony Pappenfuss on this work. So this project is a bit different in that it puts cancer into the context of the evolution of multicellularity. So of course, cancer is not just a human disease, it's observed across basically all forms of multicellular life get some type of cancer or at least some type of tumor-like growths. And this has sort of been tied to the fact that you have these mechanisms for coordination and cooperation between cells in metazoans, and when these break down, perhaps tumors can form. So sort of illustrate this concept, it's helpful to think about the major steps in the evolution of multicellularity in metazoans. And of course, our earliest ancestors were free-living single-celled organisms that had to sort of self-regulate all their processes. The first step towards multicellularity was aggregation of these single cells into colonies, which offered some protection from predation and also then allowed the sharing of resources, to the point that over time, different cells within the colony would evolve to take on different roles. And you had this process of cellular differentiation where cells differentiate and develop unique sort of phenotypes and molecular functions that allow then coordination and cooperation between cells, and for the organism then to acquire new phenotypes and take on new functions that may be evolutionarily advantageous. And this process then, is continued throughout evolution, where you get to the point where you sort of have these very highly specialized and complex tissue types and this well-defined sort of developmental hierarchies and tissue architecture that you see in modern day animals and plants. And in many ways, sort of cancer represents a breakdown of this process. But what was driving this evolution of multicellularity, what sort of and a big driver of this evolution was changes at the gene regulatory level. So when you look at the complement of genes, they're shared across species. They're actually, so many genes are very, very ancient, sort of found in multicellular and unicellular organisms. But so what has changed is sort of the timing and coordination of the expression of those genes. And so the evolution of multicellularity then required kind of new regulatory genes to emerge, new regulatory functions and mechanisms to emerge to control basic cellular processes such as DNA replication and cell division, to make sure those processes only occurred where and when they were needed. And over time, what happened is that you have these gene regulatory networks that evolved greater degrees of complexity and kind of this architecture where you have this inner layer of ancient, highly conserved genes that control core molecular processes, surrounded by outer layers of more recently evolved genes with regulatory functions that integrate signals from the environment to turn on and off these genes in the middle that are controlling these core biological processes. And in many ways, then, tumor sort of represents a breakdown of this process. It's almost like this evolution of multicellularity in reverse. So one of the earliest stages of tumor formation, you lose this defined tissue architecture, you kind of local invasion of the tumor into the out of the tissue layer and into the broader environment. Over time, these tumor cells kind of de-differentiate and start to resemble more of like a colonial mass where all the cells are kind of competing instead of cooperating and sort of regulating their own growth rather than basically listening to the signals the body's trying to send to control the timing and order of their growth. And furthermore, then even individual cells or groups of cells can break away from this mass and go seed other sites in the body. That's that process of metastasis that I was referring to. And then so this basically resembles kind of a return to sort of a free living single-celled state that is sort of dispersing out into the environment. So given these sort of parallels between the evolution of multicellularity and the progression of cancer, we hypothesized then that cancer results from a breakdown of these gene regulatory networks that evolved during metazoan evolution to control the growth and to sort of coordinate communication between cells. So we set out to investigate how gene regulatory networks formed during the evolution of multicellular get disrupted and rewired as cancer progresses. And we do this with a technique we call evolutionary network analysis. And that is that we combine comparative genomics approaches to assign evolutionary age to each of the genes in the human genome. And then we look at how does the evolutionary age of a gene relate to its position within gene regulatory networks and its propensity to have its gene expression altered or its propensity to be mutated across cancers. And so briefly, I'll detail how we kind of determine gene ages, and we do that with a technique called phylostratigraphy. And to start with, what we do is we divide organisms into different what we call phylostrata, so different levels representing sort of the main stages of evolution from single-celled ancestors all the way up to modern-day humans. And then we refer to a database called OrthoMCL, which contains protein sequence alignments from genes from 150 species. And for each of the human genes in OrthoMCL, we ask what is the most diverged species that has a clear protein ortholog to that gene. And then based on how far down the tree that organism is, we assign genes different age categories. So any gene that has a clear ortholog with a unicellular species, so a bacterium or a yeast or a single-celled protozoan, we label those unicellular genes. So we infer that that gene emerged very early on in evolution during a period of time where there are only single-celled species existing. And then the genes that we can only find orthologs within other multicellular species get labeled multicellular. And there are two categories of those genes. So there's genes that are mammal-specific, and then there are genes that are found in other non-mammal species, and those we label early metazoan because we infer that those genes emerged earlier in metazoan evolution. And so now when we looked at the gene expression levels of unicellular versus multicellular genes in cancer, we saw that many of the molecular processes that are highly upregulated in cancer, such as mRNA processing, DNA replication, cell cycle, ribosome biogenesis, those processes are predominantly comprised of unicellular genes. And in contrast, all of sort of the molecular processes that are dominated by multicellular genes, those processes by and large are downregulated across most tumor types. So what we're seeing in tumors relative to the matched normal tissues is that we see this shift towards predominant expression of unicellular genes across a range of solid cancers. So the transcriptomes of cancer cells start to look older. They start to look more primitive, more like our unicellular ancestors. Furthermore, the older genes tend to be more enriched for somatic mutations. So we did another analysis, again, dividing genes according to phylostrata, where the lower phylostrata are older genes and the higher phylostrata are more recently evolved genes. And we asked which of these, or how enriched are each of these phylostrata in recurrently mutated genes. And so our definition of recurrently mutated was either that a gene had a protein coding mutation, so a deleterious SNP or indel, in at least 3 samples per subtype. This is using data from the Cancer Genome Atlas, or that that gene was gained or lost in at least 10% of samples within a particular cancer subtype from the Cancer Genome Atlas. The results are shown in this plot, where the size of the dot is proportional to the degree of enrichment for recurrently mutated genes within each of the phylostrata. And so while some of the unicellular phylostrata are enriched for loss of function mutations or amplifications or missense mutations, what we see is the sort of strongest enrichment is within these early metazoan genes, particularly genes that kind of formed early during the evolution of multicellularity. And so it looks like the sort of selection for mutations to genes that are kind of appeared close to that boundary between unicellular and multicellular life. So furthermore, we wanted to know then, how do the genes age reflect its regulatory role in human gene regulatory networks? So to do this, we turned to annotations from the pathway commons, and we extracted out sort of all the annotations we could find on the regulatory relationships between genes. And excuse me, then we defined a set of genes as being regulators, and those are genes whose out degree in the network were higher than their in degree. So a gene's out degree is the number of genes that that it controls the transcription of. So transcription factors, for example, would have a very high out degree. They influence the expression of a lot of different genes. The in degree of a gene is the number of genes that regulate the expression of that particular gene. And so a regulator, as I said, would be a gene that regulates more genes than it gets regulated by. That was our definition. And for each of these regulators, then we could look at what were the sort of evolutionary ages of its target genes. And we could break our regulators down broadly into 3 classes. Those genes that primarily regulated unicellular genes, those regulators that primarily regulated early metazoan genes, and then what we called this unicellular early metazoan interface regulators that regulated the expression of both unicellular and early metazoan genes. And then we went back to our list of recurrently mutated genes. We asked, of the regulators on that list, how do they break down in terms of their target genes? And so we saw that regulators that were recurrently mutated through SNPs or indels, those ones tended to predominantly be these unicellular early metazoan interface regulators. So very strong enrichment for that category. In contrast, regulators that were recurrently mutated through copy number aberrations, they looked pretty similar to sort of the background of non-recurrently mutated regulators. So it really seems that to us that these kind of protein coding point mutations and short indels, these mutations that alter the protein coding sequence in tumors, there tends to be very strong selection for those when they occur in these unicellular early metazoan interface regulators. So these genes that are kind of positioned at this interface between the unicellular and multicellular parts of the regulatory network. So in our kind of model of how such mutations may lead to the development of cancer, we have a model that under sort of the steady state, you have these key regulator genes that evolved early in metazoan evolution, and they control the expression of many unicellular genes that are involved in sort of key core biological processes, such as metabolism and cell division. And they are also connected to genes, other multicellular genes in the network that sort of provide input signals to that regulator as to sort of when to turn on and off these genes. And when those the function of these early metazoan regulators is compromised through mutation or some other means, then that communication between those layers in the network breaks down. You get elevated expression of unicellular genes and decreased expression of multicellular genes. So we wanted to dig a little bit deeper into sort of how that dysregulation occurs in tumors and sort of what the consequences were for gene regulatory networks in cancer. So to do so, we turn to a technique called weighted gene correlation network analysis, or WGCNA for short. And what WGCNA does is it identifies sets of genes whose expression is correlated across a significant number of samples. And it groups these genes into what are called, co-expression modules. So this allows us to then infer how gene regulatory networks are occurring in cancer by identifying these modules of co-expressed genes. The inference being if they're co-expressed, then they're sharing sort of regulatory controls. And we could characterize these co-expression modules according to the ages of genes in the modules. So we could break them into those genes, those modules that were comprised predominantly of unicellular genes, those that were comprised predominantly of multicellular genes, and those that were a mix of unicellular and multicellular genes. And we could also characterize the network topology of these modules, whether they're enriched for certain functional groups, certain gene ontology categories. And we also calculated what we called module novelty, which is the difference, a way to quantify how different a given gene co-expression module from a set of cancer cells is from the modules that we found in the set of matched normal tissues for that tumor type. And basically, it's a count of the number of normal modules that contributed genes to that cancer module. So a low novelty module would share a lot of genes with one or two normal modules, for example, whereas a high novelty module would have genes that came from a lot of different normal modules. So genes that would not normally be co-expressed together would be the signature of a highly novel module. And we kind of saw two main patterns when we compared our co-expression modules between the normal samples and the tumors. And the first was we saw this disruption of normal modules that would get split in the tumors. So we'd see these modules that were a mix of unicellular and multicellular genes in the normal tissue, but in the tumor, the parts of these modules would be separated from each other. And this was often associated with loss of functions to these key early metazoan hub genes, these early metazoan regulators that I discussed earlier. But actually, maybe more surprisingly to us, we saw a high degree of rewiring, changes between the connections between unicellular and multicellular genes, where you'd have a module that was predominantly unicellular and a module that's predominantly normal, multicellular in the normal. These would get then merged together and to form these new co-expression modules that were a mix of unicellular and multicellular genes. And this process was often driven by amplifications, so chromosome arm level copy number gains or more focal amplifications, but the key being that genes that are sort of sharing a location on a chromosome that would not normally be co-expressed get co-expressed because that chromosome arm gets amplified. And then this can form new gene co-expression modules that can potentially confer new properties to the tumor cell. And so we called these, and these were our high novelty modules because they were quite different from what you see in the normals. So to kind of summarize what we've kind of found about disruption and rewiring of metazoan gene regulatory networks in cancer. So in a normal tissue, you have this sort of highly structured gene regulatory network where there's a lot of communication, strong links between the inner unicellular layer and the outer multicellular layer. But then sort of mutations or alterations to key hubs connecting the unicellular and multicellular layers of that regulatory network will disrupt communication between the unicellular and the multicellular genes. And then this can release a lot of these unicellular genes from repression. And this can then sort of kickstart proliferation and a lot of the molecular changes that allow tumors to dedifferentiate and proliferate and grow in an uncontrolled fashion. And this is accompanied then by not only disruption of the links between unicellular and multicellular genes, but also rewiring that creates new links between unicellular and multicellular genes that can then potentially drive further tumor progression and allow the tumor to acquire new, more malignant phenotypes. So that's the end of that section. Now I want to move on to the second project that I'll tell you about today. And that's on our efforts to develop computational simulations of clonal evolution in tumors. The work I'm going to tell you about today was performed by a very talented PhD student, Luis Lara Gonzalez, and just as a point of interest, Luis himself is actually a Mexican. He did his bachelor's in bioengineering at the Iberoamericana University in Mexico City prior to moving to Melbourne to do his master's and PhD in my group. And on this project, we collaborate with a breast cancer clinician, Shireen Loy, also a computational biologist, Tony Papenfuss, and statistician at the University of Melbourne, Howard Bondell. So some of the motivation for this project stems from the observation that can be a great variation in clinical outcomes between patients that have the same cancer type. So two patients can come in with a tumor in the same organ, and one of those patients may respond very well to therapy and survive for a long time after diagnosis, whereas another patient may relapse and decline very shortly after starting treatment. And so we were really interested in the question of what makes some tumors more aggressive than others. Is there something about the evolutionary history of those tumors, how they formed, how they progressed, how they evolved, that determines how aggressive a tumor is going to be and how bad the disease is going to be for the patient? And sort of the typical process that you go through to try to answer some of these questions is you take a sample of the tumor from a patient and you perform exome or whole genome sequencing, and then you try to identify genetic mutations, genomic alterations, and from there you try to infer the clonal structure of that tumor and sort of what events may have happened, which clones gave rise to which clones, which can be infective, but tends to be, there are gaps in that picture. It's kind of, you're trying to work backwards from a representation of that tumor at a given point in time. We decided to take a forward approach and actually simulate the evolution of a tumor from a single malignant cell all the way up to a full-blown tumor that reaches sort of a clinically detectable size. And to do this, we use what's called a branching process model. So in a branching process model, each of the tumor clones is represented as a sort of a separate entity. And we start from a single cell that we assume has sort of broken free from the constraints on its growth. So we're not modeling the process of tumor initiation. We assume that already that the tumor has initiated. There's a small population of cells that can divide and grow. And we kind of go through cycles within this model. So at each cycle, a certain subset of cells will divide, a certain subset of cells will die. And of the cells that divide, a certain proportion of those may acquire a driver mutation. And if a cell acquires a driver mutation, that enhances its division rate, increases it by a certain amount, and leads to the formation of a new clonal population. So here we started out with a blue clone, the driver mutation that forms this new green clone. The green clone will have a higher, what we call clonal birth rate, a higher division rate than its parent, same death rate, and the same mutation rate. So we have these three properties: birth rate, death rate, and mutation rate are the key characteristics of each clone. And in this fashion, we can sort of grow out a tumor in silico, starting from a single clone or a single cell. And we can track the evolution of clones, where and when they enter the population, and we can track their sizes over time. And then once we reach a size, in our case, about 4 billion cells, we have sort of a genetically diverse tumor, and we can quantify the level of clonal diversity. We can, how many clones are in there and sort of what proportion of the population they represent. So the sizes of the clones over time, they change according to these simple formulas where it's the size at the previous time point plus the number of cells that were born minus the number of cells that divide minus the number of cells that mutate and form a new clone. And the number of cells in each of these categories is randomly sampled from multinomial distribution at each time step in the model. And in this way, we can kind of track clonal evolution in silico over time as these tumors expand. And there's two key properties that we track. So we start out with a single clone that has one driver mutation. So that's what the K represents here. K equals 1. It's a one single driver, one driver clone. It will give rise to new clones that acquire new driver mutations. So in this case, the first clone that comes along is the purple clone here. So this is clone number 2. It has 2 driver mutations. And then another 2-driver clone spawns from the original clone, the brown clone here, clone number 3 with 2 driver mutations. And then it gives rise to another clone that picks up an additional driver mutation. So this is our 4th clone. It has 3 driver mutations. So in this fashion, we keep track of all of the clones and the number of driver mutations and their size over time as we grow our tumors. And this allows us to do kind of computational experiments where we vary the starting parameters for these simulations and see how that changes the results. So in our experiments, we start out with a single cell and we grow that tumor up until there are about 4 billion cells, which would correspond to a clinical, a size of a tumor in real life of about 4 centimeters cubed, which is definitely above the threshold for being clinically detectable. And along the way, we take snapshots of that population. So we record at certain time points how many cells are in the tumor, how many clones are in the tumor, the sizes of those various clones and their growth rates. And we do this we vary these different parameters that are related to the mutation rate and the effects of the driver mutations in the tumor. So there's these 2 key properties. S. So S is what we call the fitness parameter, which is how much a driver mutation increases the growth rate of a given clone. So we have both strong fitness effects, where the driver mutation makes the clone grow 10% faster than its parent, or 1% faster, or 0.1%. So we can look at the relative impact of weak versus stronger driver mutations. The other parameter we vary is the driver mutation rate, which is this parameter U, from lower to higher mutation rates. And we base those mutation rates on estimates from previous studies in the literature of the sort of frequency of new driver mutations entering the population in evolving tumors. And so we vary these parameters over these different ranges. And we perform 500 replicate simulations for each combination of parameters. And the fitness effects are calculated 2 ways. So the first way is a simple proportional fitness gain model where the driver mutation always has the same effect. So in this case, it's always going to increase the tumor's division rate by 0.11 or 10%. But we also use maybe a slightly more sophisticated or a little more realistic model that's called the stick breaking fitness gain, where the fitness effect of a driver mutation is a random variable drawn from an exponential distribution with a mean given by these values, so that different driver mutations have slightly different effects on the growth rate of these new clones. And then this actually in practice creates greater diversity. So to give you maybe a better sense of how these simulations work, I'm going to give you an example of a typical simulation and the different stages in the growth of the tumor that we see during that simulation. So for most of the simulation, the tumor size is relatively low. It's only very late in the simulation that it reaches sort of these higher clinically detectable sizes. So the first important phase is this sort of tumor initiation. For a long time, 500 cycles or more, the tumor will actually remain at a very small size. And actually we see in many of our simulations that the tumor just spontaneously goes extinct. And that's because it's a stochastic process. So just randomly more cells can die than divide at a given time step. And that can actually take the tumor size down to 0. Kind of once the tumor survives and makes it through this initiation phase, there's a certain point where then it's destined to survive and to grow out to the full size. But it does that, still does that rather slowly. It takes quite a while. In this example, another 2,000 generations. But what's key is that sort of in this initial or sort of early growth phase, that all of the dominant drivers and clones sort of the clones that are the most dominant or at the highest frequencies at the end of the simulation actually appear here. So the key driver mutations, the key clones that comprise the tumor form early on, and they actually form at a stage where this tumor would be basically impossible to detect with current sort of imaging technologies. It's just too slow. So by the time you see the tumor, sort of all its main drivers, all its dominant clones have been established. And then at some point we see this rapid size increase where the tumor kind of just turns the corner and becomes a lot bigger and rapidly increases in size. And it's also in this phase that many of sort of the resistance mutations that confer drug resistance, which we can also stimulate the formation of those. Many of these drug resistant mutations enter the population late in the tumor's evolution. And so what we see is that, again, that one of the key parameter that affects sort of the length of time it takes a tumor to grow and the diversity in that tumor is the driver mutation effect size. So tumors where the driver mutations have high impact on the growth rate, those tumors are very fast growing. They form within 3 years, and they have very low diversity, tend to be only 1 or 2 clones. Whereas when we have a much weaker driver effect size, those tumors take a lot longer to grow out, some of them over 100 years. They're very, very slow growing, to the point where they would never be clinically detected. But although they're slow growing, they have a really high clonal diversity, because a lot more mutations enter the population because it takes a lot more cell divisions for those tumors to reach their full size. And also the strength of the driver effects and the mutation rate also affect the total number of drug-resistant cells that you get in a tumor. So as the mutation rate slows down and the driver effect sizes become weaker, you actually get more resistant cells. And that's because, it takes more cell divisions. A slower growing tumor will take longer to reach a certain size. It'll take more cell divisions, and this allows more time for drug-resistant mutations to accumulate. So although the faster growing tumors may seem more aggressive, they're actually potentially easier to treat because they contain fewer drug-resistant cells. And so we wanted to actually compare how do our simulations match up with real-world tumor sequencing studies. And the issue is that, while in our simulations we can actually measure the fraction and the frequency of every single clone, in reality, when you sequence a tumor due to technical limitations and issues with tumor purity and cellular heterogeneity, you can really only detect mutations that are found in about 10% of cells. So in order to compare our simulations to real data, what we have to do is we have to provide a cutoff where we remove all the clonal populations that are found at lower than 10% frequency. But instead of actually using the clones, we actually use what's called cancer cell fraction. So we actually look at, again, the mutations. What the cancer cell fraction is telling us what proportion of cells carry a particular mutation. So we can detect mutations that are found in about 10% of cells. So in this simulation, we drop these smaller clones, and we would just proceed with the data from these 4 mutations, and from that try to infer what's the clonal history of the tumor. But obviously there's a lot of missing information. This phylogenetic tree here looks different from this one here. You've actually missed some of the evolutionary relationships. Nevertheless, we want to know sort of how did our simulations align with real world tumors. And what we found is that, again, the average selective advantage and the driver mutation rates affect the level of diversity that we see in the tumors. So Tumors with strong mutation effects and low mutation rates will only have 1 or 2 detectable driver mutations, whereas tumors with sort of weaker selective effects and higher driver mutation rates can have 2 or 3 detectable driver mutations typically. So suggesting, there's a lot of drivers in an actual tumor that are at very low frequencies that you can't detect through sort of traditional sequencing approaches. And we also see that there's a lot of variability in the level of diversity within tumors, even with the same starting conditions. So in this case, we take our tumors and we cluster them based on this cancer cell fraction. So we cluster them based on the number and frequency of detectable mutations in those tumors. And we see kind of 5 main sort of types of tumors. So we see monoclonal tumors that have 1 dominant clone, something we call dominant oligoclonal tumors, where there are 2 clones, but one of those clones is at a much higher frequency. In contrast, the balanced oligoclonal tumors have 2 clones that are roughly equal frequencies. And we also see a subset of simulations that are what we call polyclonal. So they have 3 or more detectable clones. Sometimes those clones, all those clones are at high frequency, or sometimes just 1 or 2 of those clones is at a higher frequency. And so we actually, as I mentioned, in the stick breaking models tend to see more diversity than we see in the proportional fitness model, but we do see these 5 patterns coming out across all of our subsets of simulations, though the initial starting parameters determine the relative sort of frequency of these different types of outcomes. But the message is really that you can have convergent evolution. Tumors growing under different conditions can still converge to having sort of similar properties with respect to their clonal diversity. And so then we wanted to compare our simulation outcomes to real world data. And so we came up with a method to compare the cancer cell fractions between real tumors and our simulated tumors, and basically metrics that allowed us to judge which of our simulations look most similar in terms of the cancer cell fraction, which simulations look most similar to a given patient sample. And so again, using data from TCGA, we can ask for each tumor, given its genetic profile that we've inferred from its sequencing data, which set of parameters seem to best fit that tumor. And it looks like the majority of real tumors actually fit into this category with a low selective advantage for driver mutations and a higher driver mutation rate. And this is actually closely aligns with previous studies, an early study by Bozic et al. That we've kind of based our model on, actually estimated from data that preceded the Cancer Genome Atlas. They estimated that the average selective advantage for a driver mutation was about 0.4%. That falls into our range of 0.1 to 1% for most of the tumors that we examined. And also, when we look at the best fits between TCGA and our simulations, we see that the best fits seem to align with sort of how aggressive these tumors tend to be. So the good prognosis tumors, such as prostate cancer and thyroid cancer, where patients tend to live longer after diagnosis, those types of cancers fit better to simulations that had only 2 or 3 clones. Whereas more aggressive tumors like glioblastoma and lung cancer, they fit better to simulations that had a lot more clonal diversity, 3 or 4, or even 5 or more clones. And this is in line with previous studies suggesting that the degree of genetic diversity in a tumor is proportional to sort of how aggressive that tumor is. That tumors with more sort of genetic diversity tend to have worse clinical outcomes. So just to summarize, we were able to achieve realistic simulations of tumor evolution that seem to match with observations from actual sort of clinical sequencing studies. But the evolution of tumors and the genetic diversity is heavily influenced by the impact and frequency of driver mutations, but that you can have convergence where there are different starting sets of parameters for mutation impact and frequency can result in similar outcomes. So now we're trying to investigate how these different trajectories determine disease severity and clinical outcomes by comparing our simulations to data from clinical trial patients. And also we can use our simulations to sort of look at the impacts of sampling bias, the fact that when you're sequencing, you're not seeing all the clones in the tumor. And also we're trying to incorporate non-genetic mechanisms of evolution into our model. Okay, so that's all for that section. I know I'm kind of approaching the time limit here, but I think I'll just like to quickly take you through our third project. And that is on the adaptation of prostate cancer to sustained androgen deprivation. And a lot of the work I'm going to show you today is actually being led by a current PhD student in my group, who's actually herself a graduate of UNAM. She got her master's in genetics in 2017 before coming over to Melbourne to start her PhD. So this is Rosalia Quezada Urban. Also, this work has been, some of this work has been performed by another PhD student in my group, Andrew Bakshi, who is graduating this year from Monash, and our talented postdoc Shiva. And this is done in close collaboration with the Prostate Cancer Research Program, which is spread across Monash University and Peter Mac. So just briefly, the sort of typical course of treatment and relapse in prostate cancer is shown here. So most cases of prostate cancer are actually quite benign and easily controlled with surgery or radiotherapy, and they stay localized to the prostate. But when the tumor comes back, the next line of defense is to do something called androgen deprivation therapy. And how this works is it starves the prostate tumor of the hormones that it depends on to grow. So most prostate tumors are really dependent on androgen hormones, particularly testosterone. And so there are pharmacological agents you can give to patients to sort of deplete the level of testosterone in the system to try to sort of slow the growth of their tumor. And these treatments will work for a period of time, but then often the tumor will evolve resistance to androgen deprivation. And this is what is then called castration-resistant prostate cancer. And at this point, there's no cure. You can use sort of salvage chemotherapies and stuff to try to sort of, again, slow down the growth of the tumor, but often at that point, the disease has become metastatic. And there's nothing much you can do for the patients, unfortunately. So really, we need to find new treatments for castration-resistant prostate cancer to give patients sort of options beyond androgen deprivation. And so one of the things we're doing in that regard is there's been a major effort here in Melbourne to develop patient-derived xenograft models of castration-resistant prostate cancer. And so how that works is you take a sample of tumors from a patient and you put it into immune-compromised host mouse, and in that way you can maintain the growth of these patient tumors in the lab in an in vivo environment, and that allows you to do things like detailed genomic characterization. You can expose those tumors to different drugs, see what they respond to and how they respond. And so this is soon to appear in print. We just published about 16 of our xenografts from the collection, which is part of the Melbourne Urological Research Alliance, and tissues and data from these xenografts are available for the research community. So interested researchers can contact us about getting samples to study in their own laboratories. And what particularly a point of focus in our xenograph studies is on neuroendocrine prostate cancer. So this is a rare but increasingly emerging mechanism of resistance to androgen deprivation, where the normal state of prostate tumors is this adenocarcinoma histology. But then under androgen deprivation, some of the tumors change completely to this, what's called this neuroendocrine pathology. So these cells actually change their molecular phenotypes quite dramatically under androgen deprivation. And one of the things that they do is they turn off expression of the androgen receptor. So now that they can grow, their growth is androgen independent. They don't need those androgen hormones to grow anymore. And they start expressing markers of neuroendocrine lineages such as chromogranin and synaptophysin. And these tumors are highly aggressive and very poor prognosis. And so we wanted to understand this process of evolution from adenocarcinoma to neuroendocrine and how that's driven by androgen deprivation. And fortunately for us, we have a model that, this 224 model that has a mix of adenocarcinoma and neuroendocrine cells. So in effect, we've kind of caught the tumor in the act of transitioning from adenocarcinoma to neuroendocrine. What's interesting is when you take this xenograft and you transplant it to a mouse that's been castrated, so an environment where there are no androgens, then the adenocarcinoma cells go away and you're just left with this pure neuroendocrine population. So we wanted to know what occurs in this transition. Is it just selection for the neuroendocrine cells? Do the adenocarcinoma cells transition to a neuroendocrine state? Do the neuroendocrine cells change at all? And to do this, we wanted to look at sort of the cellular level. So we actually turned to a technique called single-cell RNA sequencing. And so what this allows us to do is identify different populations within a tumor. When you do bulk sequencing of a tumor, you're kind of just getting an average expression signal, but there can be separate populations of cells that express different complements of genes. So in single-cell RNA sequencing, what you do is you take a sample, you disassociate it into single cells, and then put it into a microfluidic device where the cells get sent down one channel, and you have these aqueous droplets that contain all the sort of reagents needed to lyse those cells and make cDNA libraries that you can subsequently sequence. And so you want to get one cell into one droplet, and then those you create a cDNA library for each cell and barcode it, and that way then you can disentangle bioinformatically which genes are, or which cells are expressing which genes. And this way you can identify sort of transcriptionally distinct subpopulations within your sample. And so when we looked at this 224 intact PDX, which is the one that I mentioned has a distinct neuroendocrine and adenocarcinoma populations, we see that under single cell sequencing, when we cluster the cells by their gene expression profiles, we see a distinct cluster that's neuroendocrine and a distinct cluster that's adenocarcinoma. So the neuroendocrine cells here, the purple and blue cells, express these common neuroendocrine markers, whereas the adenocarcinoma cells express these sort of common androgen signaling markers and do not express the neuroendocrine markers. Furthermore, though, within each of these populations, we see that there is some diversity. They're not all the same. So there seem to be 5 distinct subpopulations within the neuroendocrine cells and 3 distinct subpopulations within the adenocarcinoma cells. So then we can ask what makes these different subpopulations distinct in terms of their expression profile. And so we can do things like a gene set enrichment. We can take all of the genes that are expressed in each cell and ask, are there certain groups or certain gene sets or molecular functions that are enriched in those transcripts? And in our neuroendocrine cells, we see clusters that are enriched for proliferation, so the faster growing clusters, but also other clusters that are enriched for properties like response to hypoxia and epithelial to mesenchymal transition. In the adeno, we see again a highly proliferative cluster of adeno cells, and also we see clusters that have varying degrees of expression of the androgen receptor and associated genes. Now, when we take the xenograft grown in the castrate host and perform single cell RNA sequencing, again, we see that, of course, it's just the neuroendocrine cells at this point. And this time we see 6 distinct subclusters. Again, we see proliferation and epithelial to mesenchymal transition coming up as key properties that distinguish clusters from one another, but we also see the emergence of these clusters with strong sort of invasive and stemness signals as well. So our question then is, what's changed between the neuroendocrine cells from going from the intact, where there's environment where there's plenty of androgens, to a castrate environment where there are no androgens? We want to know, so which of these sort of neuroendocrine subpopulations are equivalent across the 2 samples, which ones are maintained, and which ones are actually unique to the castrate environment. So to do that, we do something, a technique called integration, where we take data from the 2 samples and we integrate it together to try to figure out which cells from each of the 2 samples are the most similar. So the results here are plotted on this graph, where the light blue cells are the neurocells from the castrate mouse, the purple cells are the cells from the intact mouse, and then we have the adenocarcinoma cells in the orange. And what we see is that there's a strong degree of overlap between the neuroendocrine cells from the intact and the castrate host, suggesting most of these cells do retain a lot of the same molecular features after passaging into a castrate environment. And importantly, these neuroendocrine cells by and large don't overlap with the adenocarcinoma cells. So that's good, because that means our integration process is probably working properly. What we do see, though, in the castrated host is that we see a subset of these neuroendocrine cells that don't overlap with neuroendocrine cells from that were taken from the intact host. So these cells might represent sort of transcriptional programs that are only activated in neuroendocrine prostate cancer cells in a castrated androgen depleted environment. And we're following it up to more closely characterize those particular cells. So that's the end of my talk. Again, thank you for joining me today, and thank you for your time and attention. And just remains to acknowledge all the members of my lab who have contributed to these projects, past and present, as well as key collaborators and advisors who provided me with good advice on these projects over the years, and funding sources, including CAST Foundation, Peter Mac, the Australian government, and the University of Melbourne. And so thank you very much again for your time and attention, and I'd be happy to answer any questions you may have.
Thank you so much, David. Let's see if we have some questions. So if you have questions, you can raise your hands and just talk or write them in the chat. Nobody has a question. That's not.
Okay, there's a lot of stuff in there.
It's a lot of stuff, exactly. I think Mario said yesterday that he had some questions. I don't know if he's still with us right now. Oh, I think he got.
Did we lose Mario?
Is he there? I can't see it. So, well, yes, I had a question. When you do all those analyses, mostly referring to the first part of your talk, when you look at the set of genes that are modified once you see the tumors that are submitted to some kind of therapy, let me try to phrase it decently, because let me see if I can remember well what I. Before your tumors are submitted to chemotherapy, while they are not yet really recognized by the, not that they are not recognized by the body, but they are not recognized by the individual itself and by the treating physician. But there are some changes also in those signaling pathways before they are treated with chemotherapy. So how different are the changes before and after the chemotherapy?
So you mean how does the gene expression profiles of cells change?
How is the wiring different?
Chemotherapy?
Yeah.
I guess that depends on the cells and the therapy. I guess in our prostate cancer cells, we see that well, it's not a chemotherapy; it's actually a hormone therapy that's depleting the supply of androgen hormones that the prostate cancer relies on for its growth. We see that there's kind of typically two types of responses to that, and that is one is to persist in the androgen signaling pathway. So that the tumor relies on this androgen receptor gene for its growth, and that gene's activated when it binds to sort of androgen hormones. But there are ways that the tumor can either mutate or alter the functioning of that pathway to keep it going anyway, even in the absence of androgens, and make it either hypersensitive to sort of the small amounts of androgen that are left, or it can move into sort of an androgen independent pathway and start using other growth pathways to survive. And that's a process we still don't maybe understand in detail, but that's what's kind of interesting about that neuroendocrine transdifferentiation. The cells actually transdifferentiate from adenocarcinoma state into a neuroendocrine state. But then
once you know that, you can sort of predict which way the cells are going to move, because they're going to move depending on the germinal layer where they originated from. So once you know which are the ways that your tumor may move, could you anticipate a way to block that? And that would make easier an easier treatment?
Yeah, I think that's that's certainly so maybe one of the ideas or one of the goals is that if you know what cells are expressing prior to treatment, and then you know kind of which how those pathways or expression patterns enable them to kind of evade treatment and still survive, that is, then you could potentially have devise something to target those cells prior to treatment. I think that's why the single cell RNA sequencing.
The next step.
Yeah, it's a very good idea. So that's what I like about the xenograft models, is that you can actually do those experiments. You can look before and after treatment, and you can repeat those experiments, and you can see how did this tumor evolve in response to a given therapy. And so one of the things we want to do is, we want to give these neuroendocrine prostate cancers chemotherapy called carboplatin, and then do that single cell sequencing before and after carboplatin treatment to see which of the cell populations survived chemotherapy, and what molecular properties do those cells have that we might be able to target with new or existing therapies.
Right. Okay. Thank you. Mario, you had a question.
Yes, I have a question. First, I would like to apologize because I missed part of the last part of the talk because I have a problem with the power in the house. So, but finally I'm back. But so then my question is related to the first part. This multicellularity lost during the tumor evolution works fine also for leukemia?
Without applying the loss of multicellularity.
Yes, because at the end, the differentiated cells in the blood are independent. So are not in contact with other cells like in other tissues. So this multicellularity model also can operate for this kind of cancer?
That's a good question. I mean, our studies have all been in solid cancers. For that reason, we've kind of leaving the blood cancers alone. Sort of simplifies the analysis in that way. But the different sort of cell lineages in the blood, they're still part of a multicellular organism, and they still need to differentiate, and they still need to cooperate with each other. And they can't just sort of grow wherever and whenever they want. They're not totally independent either. There's growth signaling pathways that control their differentiation and growth, right? So, so in principle, all the constraints that are on a typical multicellular tissue, like the skin or the lung or whatever, would apply to blood cells as well. And so, that if they can break those constraints, though, I guess the sort of the idea is that if a cell can break those constraints that have been imposed by evolution on its growth and differentiation, then it can sort of start to grow and proliferate according to its own schedule, and in that way, it can actually gain a sort of a fitness advantage for that cell. That cell can then start to grow and divide and spread and kind of make more copies of itself than its neighbors do. And we also think that this sort of loss of multicellularity, that by sort of circumventing these metazoan gene regulatory networks, it also allows these cells to become more plastic. So they can actually enter states that a normal differentiated cell wouldn't be able to do. And so that's a feature that you see in a lot of cancer cells, that they de-differentiate, they become more stem cell-like, or they even trans-differentiate, like the neuroendocrine prostate cancer goes from being starting out as an epithelial type cell to something totally different. It becomes a neuroendocrine cell, and it looks like there's a sort of transition state that occurs, and we're trying to investigate that transition state with the single cell RNA sequencing.
So thank you. So another first question is, maybe, I don't know if you mentioned that in the last part of the talk, but it's very interesting, this transdifferentiation to neuroendocrine kind of cells. So do you know what happened with enhancers during that? The enhancers that are specific from one tissue to and then the differentiation or transdifferentiation to another one. So by, for instance, like a TAC-seq or something like that. Have you been performing that kind of studies?
No, we haven't. We would like to, definitely, with these sorts of lineage switching like that, these switching between these developmental lineages, you definitely would expect to have a lot of changes at the epigenetic level, at the chromatin level. And other studies of neuroendocrine differentiation, in which occurs also in lung cancer and some other types of cancer, have shown that there are these definitely large-scale chromatin changes, and they're probably using different sets of enhancers and regulatory elements to rapidly change the transcriptome. That's not something we've looked at yet, but I actually am really keen to start investigating that in our xenograph models. Yeah.
Mm-hmm. Okay, thank you.
Thanks.
There's another one from Alejandro Garcia.
No, but there is one in the chat too. So Alejandro, if we just can take the one question in the chat and then we take your question.
Thank you.
So should I read it then? So it's a question from Jose Leal. Would it be challenging to mix immune networks with evolution simulation to understand how the mutations will affect prognosis?
So if I understand the question, maybe asking, can we incorporate effects of the immune system on the tumor into the simulations?
Yes, so.
Which is a good question, and it's certainly the interactions between the immune system and tumors are very important for sort of controlling the growth of the tumor, and they're very relevant for understanding different clinical outcomes and what makes some tumors so aggressive. So in principle, yes, I think we're trying to build up our model sort of one step at a time, though, and just sort of it's kind of more straightforward just to model the tumor population itself. The microenvironment is obviously very important, but modeling that microenvironment can be tricky because now you've got sort of multiple cell types that you have to simulate. You have to figure out what are realistic parameters for their behavior, and you have to track them. I think where one way, though, that we could potentially model interactions with immune system or other cells is by changing those parameters of birth, death, and mutation for each of the clones according to some estimate of how many immune cells are in the vicinity. So that model all comes down to birth rate, death rate, and mutation rate. And so any sort of external factors could feed into those parameters to change the behavior of the clone. So for example, you could have a equation, a calculation for sort of the level of T cells in a tumor as a function of time. And then that could change the birth rate or death rate of all the clones in the population according to that density, or something like that. So without explicitly needing to model the immune cells themselves.
So Alejandro?
Hi, David. This is Alejandro Garcia Carranca from UNAM here in Mexico City. From your first part, can you identify particular genes? I mean, are there genes like those players in cancer like RAS or p53 involved, and so when do they came into play?
Yeah, so definitely we have seen that a lot of what we call our interface genes, the genes that connect the unicellular and multicellular layers in the network, a lot of those are canonical cancer driver genes. So p53 is actually one that fits that definition of being these interface regulators. And of course, it's very high, and one of the most highly mutated genes in cancers, and has a large effect on driving tumorigenesis. So there are a lot of other genes that came up as being interface regulators. Some of those are annotated cancer genes, and some are not. So we have that list to dig into and play with.
And for your second part, you said like those changes that improve not so much the fitness, no, of the cells are those that are favored, no? And concerning the mutation, if it's very high, the mutation level, how is it?
I see. Yes. So so I guess it's about the impact of those individual driver mutations. So so high impact mutations that dramatically increase the growth rate of the tumor clone will actually be very beneficial to that clone, but they don't create that much diversity overall in the tumor, because it's kind of like the first clone that gets one of those wins. It just gets so far out in front of all of the other cells that it can, by the time another cell acquires a driver mutation, that clone that got the first mutation is so big that the other cells can't catch up to it. So you don't see that much diversity. And you also see that the tumor rapidly increases in size, and it doesn't, there's not as much cell death, so it doesn't need as much as many cell divisions to reach that kind of that final size. And actually, because of that, it doesn't have time to acquire that many or accumulate that many resistance mutations. So although it's a very aggressive tumor and grows very quickly, it's actually relatively easy to treat. And that actually kind of aligns with the fact that a lot of sort of childhood or adolescent cancers that are fast growing are actually maybe, certain types of them anyway, like testicular cancer, are more easily controlled. Those tumors tend to be sort of more monoclonal, and they actually respond better to therapy than tumors that maybe you find more typically in older people that have a lot more genetic diversity, like prostate cancer, which is a very slow growing tumor, but it accumulates a lot of genetic mutations along the way, because it takes so many rounds of cell division to reach a certain size. And so that's maybe better modeled by the scenario where you have the very weak selective advantage, where it's only 0.1%. That's a very small advantage. So that clone doesn't really grow that much faster than the other clones around it. So they kind of have time to mutate and catch up. And so instead of sort of having one winner, you kind of have a lot more competition between these different clones. And you also have a lot more cell death in that system because they're not growing as fast. So it takes a lot more rounds of cell division to reach that sort of final size. It takes the tumor longer to grow, but because of that, it actually has more time to acquire more resistance mutations. So even though it's intrinsically a slower growing tumor, it has more diversity, and it's more likely that when you treat it, there's going to be some cells that already have a resistance mutation in there. Yeah.
Okay, thank you very nice. Thank you very much. Nice work.
Thank you, Alejandro.
So are there other questions? I mean, this is all very interesting and challenging. But then in terms of treatment, what do you anticipate is going to come out of this? Because you see that the diversity is so big and so challenging in terms of treatment.
Yeah, I think it's going to be very difficult to find one drug that would work for all the cells, for example. And then if you have, 10 or more sort of potential resistance mechanisms in there, then you can't give a patient 10 drugs. So I think what the key is actually not necessarily finding newer drugs. That's important to find some new therapies, but actually maybe smarter application of the drugs that we have. And so that's one of the things that we want to test with our model. There's this new sort of paradigm called adaptive therapy, where you don't give the patient sort of the highest dose they'll tolerate. You give them smaller doses spread out over time. And the idea is that keeps the tumor at a manageable size. Because by killing off all the sensitive cells right away, then you allow the resistant cells to grow. But if you keep some sensitive cells around, then they suppress the growth of the resistant cells.
Okay. And then combining drugs too.
Yeah, and then you can also combine drugs that way. You can sort of stagger the treatments and maybe get the tumor down to a manageable size to where then you can knock it out with a new drug or surgery. Yeah.
Okay. It's challenging, all of this. Then there's another one question on the chat that says, how would it be possible to try to reverse the tendency to unicellularity in a tumor using therapy with genes that are related to multicellularity?
Let me have a look at that. Yes, I think, what's interesting is that we, as I said, we had a lot of these typical cancer drivers come up as these genes that when they're mutated seem to perhaps facilitate this transition to unicellularity in tumors, or what we call the loss of multicellularity, really. Of course, they don't become sort of fully unicellular, but they become, they lose a lot of the features of multicellularity. So we're actually interested now in sort of digging through deeper into that list. Some of those genes are targetable by drugs, or some of them are potentially, there are drugs under development for some of these genes. So we're interested in seeing, can we find a way to use that information about a gene's age and where it is in a regulatory network to try to prioritize it as a drug target for a patient that carries a mutation or alteration to that gene.
Okay. Well, I think, thank you. So I think we are, I don't see if there are any other questions. Do you see any other questions, Mario?
No.
So, yeah, we would really like to thank you, because this is very, very interesting, and challenging in terms of how we think about cancer and how curable cancer will be.
Yes, thank you. Well, it's been a great experience being involved in this talk. So it's been great meeting you and all the students. And I think I've really learned a lot from your questions and feedback as well. So thanks very much for inviting me to be a part of this.
You were meant to participate. You had too many Mexicans in your lab already.
Well, I want to say that personally, I think, you do a great job of training your young scientists. I actually have one, I just took on another Mexican student from Monterey, and I really enjoy working with the Mexican students. I find that they have good technical proficiency, but also that ability to think in the abstract and think creatively. It's really a great combination, and of course they have a tremendous work ethic and good attitude.
Yeah,
so you guys
we're happy
are
to know that
young scientists there.
We're happy to know that, and we would also be very happy in the future to be able to offer jobs for them, because that's our problem right now.
Yes. Yeah, I've had those discussions where unfortunately some of them feel like they have to leave Mexico for more opportunities.
Exactly, and there aren't that many jobs openings, so that's really sad.
Yeah.
We would like to take advantage of the preparation and and how smart they are. I agree with you.
Yeah. I don't know. Do you have any is it possible for joint appointments where people could sort of have jobs where they work at a university in Mexico and in another country
and in Australia, for example? It's not so frequent, but it would be nice if we could develop that kind of new concept in terms of joint appointments and how people could be able to work into different parts and benefit the two different places where they are working.
Yeah, definitely.
So, well, thank you so much again for joining us, and I would really appreciate you giving us two half days.
Oh, you're welcome. You're welcome. Thanks again for the invitation, and I really enjoyed being a part of your program.
All right. Thank you so much.
Okay. Thank you very much, David.
Thank you.
Thank you so much again.
Okay. Probably I will contact you. Oh, over here really close. That's okay. Ya se salió.
Ya se salió. Bueno, ok.
Oh, les digo. Ok.
Bueno, pues muchas gracias a todos por estar con nosotros.